Okay. Hello, everyone. I am Yunzhuo Zhou, or Emily, and I'm from Professor David Asher's lab in the School of Chemistry and Molecular Biosciences at the University of Queensland, Australia. And today I'm going to present my work, DDMut-PPI, predicting effects of mutations on protein-protein interactions using graph-based deep learning. Protein-protein interactions are physical contacts of high specificity established between two or more proteins and represent complex biological functions. The interactions can happen between enzyme and regulators, in this case between CDK2 and cyclin A to control the cell cycle. The interactions may also provide structural support, for example, holding the collagen molecules into this triple helical structure. And also the interactions between antibodies and antigens can help humans to fight against the invading pathogens, and the recruitment and interactions between transcription factors can control the gene expression level. The interface residues at the protein-protein interaction sites are particularly important because they can control the specificity, strength, and the regulation of the protein-protein interaction. Usually, the interface residues are defined as those having at least one atom within a 5 angstrom distance to any atoms in the opposing protein chain. And it has been shown from a study that somatic missense mutations are significantly enriched in PPI interfaces compared to non-interfaces in more than 10,000 tumor exomes. So studying how these mutations would affect the protein-protein interactions may help researchers to better understand the molecular mechanisms underlying the disease and possibly developing new strategies to targeting these protein-protein interaction sites. The effects of missense mutations on proteins can be characterized as a thermodynamic cycle. When a protein folds from the 1D sequence to the 3D structure, it releases the Gibbs free energy, delta G folding. And the difference between the delta G folding for the wild type and the mutant determines how the mutation would affect the protein stability. In my previous work, I developed a tool called DDMut to predict how the mutation affect the protein stability. And I also presented this work last year in Lyon. And now I'm expanding this thermodynamic cycle and trying to predict how the mutation would affect the protein-protein interactions. So when a protein A binds to another protein B, it releases the binding free energy, delta G binding, and the difference between the wild type and the mutant determines how that mutation would affect the protein-protein interaction. And a value smaller than 0 means the mutation is decreasing the affinity, and a value greater than 0 means it's increasing. So here I developed DDMut-PPI, a tool to predict the delta-delta G binding, and this work has been published in Nucleic Acid Research earlier this year in the web server issue. First of all, I want to introduce the data set I used. The training data set was from ScanPy2. This is a widely used training data set for almost all the delta-delta G binding predictors. However, within the data set, there are much more mutations with delta-delta G values less than 0. They are decreasing the affinity. And in order to balance this delta-delta G distribution, we introduced the hypothetical reverse mutation. So theoretically, when an amino acid changes in the reverse direction from the mutant to the wild type, the delta-delta G would be equivalent to the negative delta-delta G of the corresponding forward mutation. So we can see after including these reverse mutations, in the overall data set, the delta-delta G follows a normal distribution with a mean value of 0, and it has equal number of mutations decreasing and increasing affinity. This is the overall methodology workflow. The delta-delta G values were from ScanPy2 and also from several blind tests are from several other literatures. And the protein structures were just downloaded from Protein Data Bank. And then we generated a set of features capturing both the mutation sites and also the overall protein-protein interaction interface. The substitution matrix account for the amino acid changes to capture the physical chemical changes brought by the mutation. And we also calculated atomic interactions, the energetic terms from fold X, and also the pharmacophore changes. And we also included another set of features named graph-based signatures, which capture the distance patterns between pairs of atoms within a local 3D environment. And I will discuss this in more details in later slides. The deep learning architecture is essentially a Siamese network with a forward component for the forward mutation and a reverse network for the reverse mutation. And this is combined with a graph convolutional network operated on the protein interaction interface graph. And I will also discuss the architecture in more details in later slides. And eventually this model was deployed to a web server, and it's freely accessible by all the users. In order to better capture the interaction patterns at the interface, we modeled the local residue environment using atom level graph. And this graph is the basis where we extracted the graph-based signatures. So within a 10 angstrom distance from the mutation site, all the atoms within that environment are modeled as nodes. And there are two types of atoms. So the purple color indicates the atoms within the protein where the mutation happens, and the green atoms indicates the atoms in the opposing protein chains. And we draw the edges between atoms based on their distances from a minimum of 1.5 angstroms and increasing with a step of 0.5 angstrom and until it reaches the maximum 10 angstrom. So there are within protein connections and also between protein connections indicated by solid and dashed lines. And also each atom is labeled with one of the 8 pharmacophores shown here. And in the end, we would get a cumulative distribution of different types of edges. So that is different pairs of pharmacophore counts and against different distance thresholds. Apart from the atom level graph, we also modeled the interface interaction using residue level graphs. So here, each node is an interface residue, and we annotated each atom using the protein file embeddings from the ProChance protein language model, and each edge is an interaction calculated by a program called Arpeggio. So it has different types of interactions such as aromatic, hydrophobic, hydrogen bond, and so on. And in this way, we can sort of combining the evolutionary information with the interactions patterns at the interface. And the detailed DDMut architecture is a Siamese network, the forward network for the forward mutation and the reverse network for the reverse mutation. And they share exactly the same weights. And we used a special loss function inspired by our previous work. When we're minimizing this loss, we does not only optimizing the model accuracy, that is the prediction for the forward mutation is approaching the experimental value y. But at the same time, we are also optimizing on the model antisymmetry, that is the prediction for the reverse mutation is approaching the negative prediction for the forward mutation. So both the first and the second terms are approaching zeros. This is the detailed architecture for each subcomponent. The graph-based signatures extracted from the atom level graph I mentioned previously were processed by convolutional network and transformer encoders. and all the other complementary features were processed by 2 dense layers. And there's a graph convolutional network operated on the protein interface graph, and they all concatenated together, followed by another dense layer, and gives the predictions. This is the performance of the model on predicting the effects of single point mutations. Under LIV1 binding site out cross-validation on the ScanPy2 dataset, the model achieved Pearson's correlations of 0.67. And here we divided the full ScanPy2 dataset into 138 folds, depending on the holdup type annotation from the ScanPy2 database. So these are essentially different types of protein interactions, such as protease inhibitors, antibody antigens, and so on. And at each time, we only tested on one of the interaction type and trained on all the other types. And we also found that after removing the 10% outliers colored in red, the performance improved significantly to 0.79. And we also compared our model with several other methods on both the ScanPy2 training data and on 3 different blind test sets. S645 contains only the antibody antigen complexes, and these 2 are deep mutational scanning data set for spike ACE2 and MDM2P53. And across all the data sets, DDMut-PPI achieved comparable or better performance with the top 2 methods for each data set. In order to determine whether all the architecture components are actually working and contribute to the final prediction together, we did an ablation study. So we found that by disabling the graph-based signature part relies on the atom level graph, the performance drops to a larger extent, and by disabling the other features or the graph convolutional network part, the performance also drops, but to a lower extent. And here's the performance on predicting the effects of multiple point mutations. Since we didn't have much data point for the multiple point mutation, instead of training a separate model, we just summed up the prediction for each single mutation within that multiple point mutation from the single mutation model. And even with these, the model achieved Pearson's correlations of 0.83 on a dataset SM1124. This is a training dataset used by another method. And we also compared our method with several other methods to other blind test sets, and it shows comparable performance. This is the web server interface. On the submission page, users can submit a single point mutation by providing a wild-type structure, specify the mutation details, or uploading a list of mutations. And users can also perform interface analysis or predict the effects of multiple point mutations. This is the result page for single point mutations. It shows the predictions, the delta delta G values, and shows whether it's decreasing or increasing the affinity. And it also shows the predictions for the reverse mutations. And within the interactive viewer section, users can visualize the 3D structure, the interactions, and also rotate the whole structure. And for the interface analysis, there are 2 different modes. The first mode is alanine scanning. So basically, it mutates each single interface residue to alanine and shows the results as a bar chart, and then map the predictions onto the 3D structures. And under another mode, it outputs the results for saturation mutagenesis. So it mutates each single residue to any other possible residue and shows the result as a heat map. And in this way, users can visually see which interface residues are actually contributing more to the protein-protein binding. In conclusion, DDMut-PPI is an accurate tool to investigate the effects of missense mutations on protein-protein interactions. And the deep learning model was built by integrating graph-based representations of the localized 3D environment with the prototype protein language model embeddings, leveraging both forward and the hypothetical reverse mutations to account for model anti-symmetry. And it outperformed other tools on several blind test datasets and is freely accessible as a web server and an API at the following link. In the end, I want to acknowledge the University of Queensland, Baker Heart and Diabetes Institute, the Australian Government Research Training Program, and my supervisors, Professor David Asher, Yu Chang Myung, Carlos Rodriguez, and I also want to thank all the other lab members for support. Thank you.
Thank you very much. Do we have any questions?
Hi, excellent work. I was wondering on the heat map of the saturation mutagenesis, what are the black cells?
The black cell is just the amino acid itself. For example, it's alanine on the position, for example, 200, and you mutate into the other 19 possible residues.
Can you show it, please? Could you show the slide, please?
Oh, okay.
Because I thought that's what it was too, but I've been looking at it, and that last one on the corner, it's alanine, and the position is an E. So.
Here, the first letter, it means the chain number, not the original residue itself. So it means chain E, position 152.
Right. Okay, thank you.
All right, we have time for one more question.
Great talk. Great talks. Just wondering, do you think you can extend your model here for the SNPs to the small indels?
Indels, I mean deleting the...
Yeah, like a small deletion.
Small deletions. And then you mean the protein will have missing residues or?
Yeah, because for example, you can think about it, like general CRISPR editing usually will create some kind of small deletion. So it will be interesting to see how that impacts the protein PPI interaction.
So for me, the chaining sets only have missense mutation, so just single amino acid change. So we didn't account for any indels.
Do you think your framework can extend to that?
If we have new chaining data, it's possible.
Okay, thank you.